Welcome to Unit 3, Video Lesson 2. Today we're going to be talking about Part 1 of a three-part story. We're going to be discussing DNA replication. And as you can tell, the cartoon is about duplication, and replication is just another word for that. So by the end of today, you should be able to briefly describe the differences between replication, transcription, and translation. And that's what we're going to cover in Part 1 of video, which is mandatory. And then in class, we're going to go over replication, including identifying why do we do it, how is it done, and who's involved, in particular what enzymes are involved. So Part 2, video, is optional, as this will actually be done in class. So before we begin, we're actually going to talk about a three-part story where we can have the option to go from DNA, Part 1, back to making more DNA, a process that's called replication. Or where we can go from DNA to make mRNA in a process that's called transcription. And then finally, from mRNA to protein in a process called translation. So that's the three parts of our story. One is replication, which is just to make more DNA. And Part 2 and 3 are transcription and translation. And their goal is to actually make a functional protein. Now, we're going to teach these in order of replication to transcription and translation. And because the words are very similar, they're often confused. And one hint I'd like to give you, we actually learn the processes in alphabetical order. So if you can remember what order you learned the process, but you're confusing the names, just put the names in alphabetical order. Okay. So last day we were looking at DNA pretty much from the atomic level, very microscopic. Today I'd like to give you a chance to consider at a slightly bigger level. So we'll start with what's happening in the nucleus, and you have these nice little X-shaped chromosomes. So what is a chromosome actually? Chromosome is a tightly wound up, we'll say, chunk of DNA. It's the way our body keeps it nice and neat. Okay? And in terms of chromosomes, humans have 23 pairs of chromosomes. So we have 23 separate chunks, of which we have two copies of the pairs of chromosomes. And why do you think I put pairs in quotes? What we really have are 22 autosomal pairs, and these are identical pairs. And then we have one pair, which in females is a true pair, one pair of sex chromosomes. So that would be our XX in female and your XY in the male. And that's why it's not really a true pair, because it actually has one different from the other in the male. Okay, autosomal just means anything that isn't related to the sex chromosome. Okay, and if we think about the pairs, where are the pairs coming from? One from mom, your biological mom, and one from your biological dad. And while the chromosomes are said to be identical, they're not 100% identical. The one from mom and the one from dad, they'll both have genes that code for eye color, but that specific color will be different from mom to dad, potentially. And when we look closer at a chromosome, and we'll kind of zoom in, that's what this picture is doing. It's starting at the larger scale and then zooming in. We have an X shape, and chromosomes truly are X shaped, and they have, in this case, absolutely, barring a mutation, identical sides. So that's what we call sister chromatids. Equal two identical copies of the DNA. And we're going to talk about why we need those two identical copies down lower, and then we'll talk about how our body goes about replicating to create those two identical copies. Okay. If we zoom in even farther, we get to our more traditional DNA double helix. And when it's unwound, which is necessary for copying, imagine someone hands you this crumpled up ball of notepaper with writing on it and tells you you need to copy it. The first thing you're going to do is unwind it. So when DNA wants to make a copy, it unwinds the chromosome, and when it's unwound, it's called chromatin. And in its unwound form, a DNA double helix, you go across the ladder from side to side. You remember that's a purine with a pyrimidine, is only 2 nanometers wide. And if that's hard for you to understand, just how small is 2 nanometers? It's 2 times 10 to the negative 9 meters, or 0.8 zeros followed by 2 meters. But I can do the obligatory, oh wow, that's really small, but it still makes it hard for me to understand. The easiest way I can understand how small this is if you were to take a dime and look at it from its edge. That's about approximately 1 millimeter thick. And a dime edge could fit 500,000 or 500,000 strands of DNA across it. If we put it the ladder, like ladder to ladder to ladder, you could fit 500,000 or 500,000 across just on the edge of the dime. And what makes that so impressive is that tiny thing. When the egg and sperm came together to make you, there was only 23 pairs of those, 46 chromosomes that could fit in that tiny space, and that turned into you. That incredible tiny little amount of information became what is now you. That's pretty impressive. Okay, so we're just gonna stop there pick up the rest in class. If you have any questions about this, don't hesitate to ask in class. Otherwise, I'll see you there, and have a great day. Thank you.